Hi, today I'm going to talk about a paper of title Microbes and Alzheimer's Disease. A worldwide team of senior scientists and clinicians have come together to produce an editorial which indicates that certain microbes, a specific virus, and two specific types of bacteria are major causes of Alzheimer's disease. Their paper, which have been published online in a highly regarded peer-reviewed Journal of Alzheimer's Disease, stresses the urgent need for further research and, more importantly, for clinical trials of antimicrobial and related agents to treat the disease. This major call for action is based on substantial published evidence into Alzheimer's. The team's landmark editorial summarizes the abundant data implicating that microbes, but until now, this work has been largely ignored and dismissed as controversial, despite the absence of evidence to the contrary. Therefore, proposals for the funding of clinical trials have been refused, despite the fact that over 400 unsuccessful clinical trials for Alzheimer's disease based on other concepts were carried out over a recent 10-year period. Opposition to microbial concepts resembles the fierce resistance to studies some years ago which showed that viruses cause certain types of cancers and that a bacterium causes stomach ulcers. Those concepts were ultimately proved valid, leading to successful clinical trials and the subsequent development of appropriate treatments. Sterile red blood cells were seen to contain dormant microbes, which also had implications for blood transfusions. There is evidence that Alzheimer's disease has a dormant microbial component and that this can be woken up by iron dysregulation. Removing this iron, which slows down or prevent degeneration, they can't really ignore all these evidences. The microbial presence in blood may also play a fundamental role as a causative agent of systemic inflammation, which is a characteristic of Alzheimer's disease, particularly the bacterial wall, cell wall component, and endotoxin lipopolysaccharide. Furthermore, there is an ample evidence that this can cause neuroinflammation and amyloid beta plaque formation. The findings of the editorial could also have implications for the future treatment of Parkinson's disease and other progressive neurological conditions. Many studies, mainly on humans, implicating specific microbes in the elderly brain, notably herpes simplex virus type 1 HSV1, chlamydia, pneumonia, and other types of spirochetes in the etiology of AD. Fungal infection of AD brain has also been described, as well as abnormal microbiota in AD patient blood. The first observations of HSV1 in AD brain were reported almost 3 decades ago. The ever-increasing number of these studies, now approximately 100 studies in HSV1, warrants re-evaluation of the infection and AD concept. AD is associated with neuronal loss and progressive synaptic dysfunction, accompanied by the deposition of amyloid beta peptide, a cleavage product of amyloid beta protein precursor, and abnormal forms of tau protein, markers that have been used as diagnostic criteria for the disease. This constitutes the hallmark of AD, but whether they are causes of AD or consequences, it is still unknown. It is suggestible that these are the indicators of infectious etiology. In the case of AD, it is often not realized that microbes can cause chronic as well as acute diseases, that some microbes can remain latent in the body with the potential for reactivation, the effects of which might occur years after initial infection, and that the person can be infected but not necessarily affected, such that controls, even if infected, are asymptotic. Thank you for watching the video. If you like the video, don't forget to like and follow my page so that you can get other updates in the future. Thank you.